Hi, my name is Mark Bonizzoni. I'm an associate professor in the Department of Chemistry and Biochemistry at the University of Alabama. I want to welcome you to our virtual visiting session, and I wanted to give you a bit of an idea of what we do in the Bonizzoni group at the University of Alabama. So for a start, I wanted to share with you some of our favorite things in the group, and those are water-soluble polyelectrolytes. So polyelectrolytes are polymers that contain charged groups, either on their surface or in their chain. So for instance, you can have polymers like these that are more or less what you would be used to, linear polycations or polyanions that just have positive or negative charges on their surface or along their chain. But you also can have branched structures that are maybe less common as polymers. They start out from a core and they develop in all directions. And typically these are three-dimensional structures. They're pretty common, in fact. So you can see these kind of polyelectrolytes, both branched and linear, used in petroleum refining, used in food technology, used in drugs and drug delivery. Our group is interested in supramolecular chemistry, both from the organic chemistry standpoint and from the analytical chemistry standpoint. Now, to give you an idea, supramolecular chemistry has to do with the chemistry of non-covalent intermolecular interactions. So non-covalent means we're talking about hydrogen bonding, electrostatics, hydrophobic interactions, and so on and so forth. Intermolecular is the way the molecules recognize and act and talk to each other. So we're typically interested in chemical interactions, and we tend to reduce those to artificial systems because otherwise these interactions are pervasive. They're everywhere, for instance, in living organisms. They're the very basis of biology and biochemistry. But we are interested in carving out that part of the world that has to do with artificial systems that are designed to display these interactions. The field has been recognized before in 1987 with the Nobel Prize to Cram, Land, and Pedersen for their work in fundamentally founding the field and inventing the whole idea of supramolecular chemistry. And again in 2016 to Savage, Stoddard, and Thringa for the pinnacle of its development in a way to get into the point of molecular machines and devices. In our group, we have started out and have been very interested for a long time in polymers that are called PMM dendrimers. So these are polyamidoamine polymers that are hyperbranched polyelectrolytes. They start from a core here in pink, and they grow out in all directions by branching every time you encounter a tertiary amine. And you can see that you have an amine, amide, amine, amide. So you have an amido-amine building block. And we use these polymers in different ways. So on the one hand, we've done quite a bit of fundamental studies on them from the physical organic side of the house. We looked at the key interactions that make these polymers recognize smaller molecules in solution. We asked ourselves essentially how these polymers work in order to behave as supramolecular hosts. We've done that by doing studies, typical physical organic studies, say calorimetry and spectroscopy. But we've also done synthesis of model compounds and studied the model compounds separately to determine the fundamental interactions that are at play. And when we learned something, we applied it to the analytical side of the house. Essentially, we continuously used a feedback loop, if you will, in which we developed new devices using these polymers, and then we understood how the devices work using the physical organic methods, which allowed us to develop newer and better devices for analytical and bioanalytical environmental purposes. And you can see here in this spread, there's been a number of people over the years, a number of graduate students and undergraduates that have helped me out in this pursuit. One of the most interesting properties of these polymers is the fact that they allow you to construct multivalent interacting systems. So what I mean by that is the fact that, for instance, in these polymers, you have multiple binding sites, multiple interaction sites on the surface of the polymer that are very close to each other, typically adjacent, so that they can get you possibly increased affinity and possibly cooperativity of behavior. That is, one group binding helps the second group binding to the same host. And here you can see two possibilities of the non-cooperative here in the left. Your guest binds one first time and the second guest binds just another position, versus a cooperative behavior. The binding of the first guest makes the binding of the second guest more likely. Additionally, we have also found that these are very useful to confer water solubility to things that otherwise would not be water soluble at all. Here's an example of how we use synthetic organic chemistry and polymer chemistry to achieve an analytical purpose ultimately. So here we started out with a PAM-AM dendrimer that is bristling with primary amines, and we added some boronic acid functionality, which we know from previous experience in literature binds the sugars very well. And so we were able to do this in a completely tunable fashion. So we developed synthetic methods, and we developed methods, spectroscopic methods to monitor the system, so that we could essentially dial in the amount, the number of substituted sites we would have on the surface of these dendrimers, with almost complete control, all the way from minimal substitution to maximum substitution that would give us insolubility in some cases. And then we combine these with very simple organic dyes in a system that is called an array sensing system. And we developed such an array sensing system for analytical purposes, mostly for biological and environmental purposes, to sense simple carbohydrates. And in fact, we expanded it to more complex carbohydrates as well. So essentially, the system was able to tell which carbohydrates it was exposed to, and in some cases at what concentration as well. And this was very simple, because very, very powerful as well, because the simple boronic acids would not have been able to do that in water at neutral pH as this system was. The extra features, the higher affinity, the water solubility and compatibility, completely came from the polymer. The really interesting question here from the physical organic standpoint is the background. So analytically this works, and that's great. But from a physical organic standpoint, we're very interested in understanding to which extent cooperativity increased the binding affinity in these systems, and also to which extent we can switch on and switch off cooperativity, and in what conditions we can do that, and how that depends, for instance, on substitution density at the surface. And we were able to access these properties because we had a fully tunable material, so we could actually make different substitution density materials and study them separately, and arrive at interesting fundamental conclusions on the way that the binding works. I've kind of introduced this, slipped in a little, the idea of array sensing. So array sensing is the way that we mostly use these polymers from an applicational standpoint. We turn these polymers into sensors by congregating multiple ones together and making them work together. And the idea of array sensing is simple yet powerful. If you have a sensor of an array of relatively bad sensors, not particularly selective ones, they may still be able to work together to give you sensitivity and selectivity, because you may be able to expose them to different analytes, and each analyte may give you a characteristic fingerprint or a pattern of behavior in their interaction with each one of the different sensors in the array that is characteristic, and that gives you back selectivity where you had none for the simple arrays. This has been very, very successful in our hands. So we've been able, for instance, to work on nucleoside phosphates all the way to organophosphates. This was published in JAX a few years ago, to carbohydrates, both simple and more complex carbohydrates, as I hinted at before, to metal ions, and for instance here to carboxylate anions on solid support on disposable paper strips, and to other systems as well. So this is just to give you an idea of the generality of the approach. We can actually use this approach to tune it to almost any analyte that we're interested in, and we can use it in a modular way by constructing whichever sensor we need at the time, starting out with a polymer, decorating the polymer in different ways to give us an array of sensors, using the array of sensors to do fingerprinting to give us analytical differentiation, analytical power. It would also be interesting to construct the array of sensors directly on the polymer. This will require a relatively more refined and more complex organic/polymer synthesis, and I would be delighted to attract students to work on this project among others that will combine uniquely polymer chemistry, synthetic organic chemistry, and analytical chemistry as well. And also always with an eye out for the fundamental science behind it and the studies of cooperativity. This is by no means the only thing that we're working on, but it's definitely one of the interesting and underutilized openings in my group. So generally speaking, in my group, what would you mostly be exposed to? Well, we're pretty interdisciplinary, as we talked about. So you'd be coming in, for instance, to work on synthesis and polymers, and as an organic chemist, or on chemical sensing and instrumental work as an analytical chemist, or on calorimetry and spectroscopy as a physical chemist. And I've had students from, at this point, actually, all walks of life walk into my group and find their niche and find their special combination of topics and skills that best attracted their interest. We're heavy users of organic characterization techniques, obviously, because we do have synthesis interests, and we also have interest in characterization from a physical organic standpoint. So we're heavy users of mass spec. We have this new, very shiny spaceship-like toy as a multi-toff that was bought thanks to NSF funding. We have recently also received funding as a group of PIs to purchase a new NMR with a cryoprobe that is being installed these days. And we also have specific techniques to our lab. So we're very heavily involved in optical absorption and emission spectroscopy. We use high throughput techniques, microwell plate readers, and so on. And we also have an isothermal titration calorimeter that is showed over here, in addition to this mini tau as a lifetime fluorescence emission machine. So we have a somewhat unique combination of organic synthesis and instrumental techniques in our lab. I also pride myself in my students' ability to disseminate their work and in networking. And one example here is the fact that my students, these are three of my students. This is a collaborator from the University of Southern Mississippi, Dr. Joshua Trope. They organized a student-led symposium that was selected for support at the ACS national meeting in 2018. They organized the whole thing. They contacted speakers, made the schedule, found funding. It was an extremely interesting and powerful opportunity for them to network and get exposed and gain important experience in the world of scientific dissemination More commonly, we would be found disseminating our results at the Southeastern Regional Meeting of the ACS, CERMAX, ACS national meetings, the physical organic research conferences. I tend to go to the physical organic ones and the polymer ones, and in water symposia, national and international analytical symposia similarly. And we've published more or less far and wide, both from the supramolecular specific to analytical to general journals of high impact, like the Journal of Chemical Society, of the American Chemical Society, Chemical Science, and Analytical Chemistry, and many others. Finally, I just wanted to mention our current funding that is provided mostly by NSF through an EPSCoR research funding on deployable polymers for Gulf of Mexico applications, instrumental support for the purchase of the multi-Toft-Off spaceship that I showed you before, and for a purchase of a new cryoprobe NMR spectrometer. I would also point out that we are a very collaborative group. We have standing collaborations with Professor Jason Azalea at the School of Polymer Science at the University of Mississippi, Professor Eugenia Carlampieva at the University of Alabama, Birmingham Department of Chemistry. And over the years, my students have had support from the Department of Education and NSF as well. So in short, I just wanted to thank you for your attention, remind you that I would be more than happy to answer any of your questions. If you have any, please shoot me an email, or we may have a chance to meet during our live event coming up this week. Thank you for your attention, and I'll see you soon.